Hey guys, my name is Max and welcome to Simple Biology. In this video we're going to be talking about mitosis. Now mitosis is a part of the cell cycle, and I talked about the cell cycle in my last video and I mentioned mitosis briefly. We're going to go into a little bit more depth because mitosis is just incredibly important to life. It's the process by which a cell will divide in order to form two genetically identical daughter cells. And so this is the way that the cell is going to pass on its genome to the next generation. So there are a couple of we're going to go through all the phases of mitosis, but first we're going to start with the G2 phase, which is actually just prior to mitosis. And a couple of characteristics of G2: it's still got the nuclear membrane, the nucleus is completely intact. It's got nucleoli, or just one nucleolus in this picture. The DNA is uncondensed, just kind of all over the place, and there are two copies of the entire genome. If you remember, during the S phase of interphase, the genome was copied. S is for DNA synthesis. So now we have two copies of the genome, which is going to be important as we go on into mitosis. And you also notice that there are two centrosomes. Each centrosome having two centrioles, meaning 4 centrioles in total. Because there's two centrosomes, one is going to be passed on to each cell, which is going to be also important. As we start mitosis, the first phase is going to be called prophase. Pro means like before or like prior to. So prophase is the first phase of mitosis, and we start to see that the nucleus begins to break apart. The nucleus in this video is going to be represented by these little purple lines, and so the nucleus has just fragmented. The nucleoli disappear completely. And so you can see that they're no longer present, and those chromosomes start to condense. Now you can view them as discrete chromosomes instead of just a massive chromatin. In addition to this, the sister chromatids, meaning two identical chromosomes, will attach to each other and form what's called a kinetochore right there. It's that protein part in the middle of the two chromatids. In addition to this, those centrosomes form the mitotic spindle. It's named just because of its shape, but essentially the mitotic spindle is a bunch of microtubules that extend throughout the cell, starting in those centrosomes. There are going to be two types of microtubules: kinetochore microtubules, which will attach to the kinetochores, as their name suggests, and polar microtubules, which extend and eventually attach to other microtubules. Also, it's going to be important to know that there's a type of protein called a cohesin, which attaches the two sister chromatids. Cohesin, I guess, but that's the protein that attaches them. The next phase of mitosis is called metaphase. Now, the way I always remember this is that the M in metaphase is also the first letter in middle. And as you can see, a very strong characteristic of metaphase is that all the chromosomes are right in the middle of the cell. So the chromosomes have light up at the center of the cell, and this imaginary line is known as the metaphase plate. This is eventually going to be where the cell splits in half. In addition, by now the centrosomes are at opposite ends of the cell, and so the mitotic spindle is extending throughout the cell. It's important to know that metaphase is actually the longest stage of mitosis, because throughout metaphase these microtubules are all getting into place, and metaphase cannot end until every single microtubule is in place. Sometimes that means several different microtubules at each sister chromatid. So you can imagine that it would take a long time to get that all organized. Once metaphase is complete and all those chromatids are all in the right place and the microtubules are in place, the cell goes into what's known as anaphase. Now, in anaphase, those cohesins that I mentioned earlier are cleaved. That means that the chromatids are no longer attached to each other and are free to separate. The cell starts to break apart some of those microtubules and pull each chromosome. It's now a chromosome because they're separate. Each chromosome is pulled by that kinetochore and the kinetochore microtubules, which are shortening. The cell begins to elongate, and form less of a spherical shape and more of a oval shape. And this is the shortest stage of mitosis because it doesn't take quite as long for them to just get to the end of the cell. And once they're in place, which is rather quickly, all these microtubules can start to break down and go into the next phase. This last phase is known as telophase. Telo meaning the movement or change like that. So as you can start to see, all of these chromosomes are moving even further towards the right locations. The mitotic spindle disappears, as you can see, and the centrosomes are just kind of sitting there, ready to perform their function in the newly formed cell. Nuclei reform in each cell. This is where you can start to see the two distinct cells, and those chromosomes uncoil. Also, the nucleoli reappear, or one nucleolus each in this case, and the cell starts to divide in two. Lastly, we have cytokinesis. Now, cytokinesis is not actually part of mitosis, but it's the process by which the cell actually pinches into two. It starts during telophase. It begins during telophase, and I showed you right here where the cell starts to pinch. That's cytokinesis right there. It's not actually part of mitosis, but it happens at the same time. In animal cell, this is known as a cleavage furrow, and it's just a bunch of protein complexes that tighten and tighten until the cell pinches right into two. On the other hand, when you have plant cells, they form what's called a plant cell. And what this is that a bunch of vesicles will start to line up in the center of the cell and fuse until the cell has formed a completely new, or the one cell has divided into two completely new cells, and cellulose will start to form in that gap between them. And so that's really it for mitosis. It's as simple as that.